So today we're going to start off directly with some content to make sure that we can get into clotting by the end of today. So today we're going to start off directly. Sorry, I can hear my live feed lagging a couple seconds behind me. I'll close that out. Okay. All right, we want to get to clotting by the end of today because clotting is super interesting, super important, but also kind of complicated. So let's get ourselves set up. Okay. I'm gonna put the chat over here. And anybody comes in late, I'll see them over there. Okay. So we're continuing with our cardiovascular system. And when we left off, we had been talking about the synthesis of the erythrocytes. So let's pull those up. Okay. So we had looked, I'll skip us back. The last slide we had looked at kind of looked like this. We were looking at how erythrocytes, after they start to differentiate, so after they start to actually become red blood cells and split off, they go through this pathway. They start to lose their organelles, they lose their nucleus. During this time, they are producing hemoglobin. And then eventually, they're basically nothing but a bag of hemoglobin and carbonic anhydrase carrying around oxygen and carbon dioxide for us. Okay. So now we're going to pick up thinking about the requirements for that synthesis. And basically, we need some components able to allow us to produce our red blood cells. So one thing we need is iron. Iron is a key component of hemoglobin, right? That heme portion in the center of each of those 4 subunits of our hemoglobin, that heme portion contains iron, and we need it to carry oxygen. So there's no point in making red blood cells if you don't have iron. And we're going to be tracking how iron actually gets reused when we are digesting and then remaking our red blood cells because it's so critical to their function, critical to their whole role. So the normal hemoglobin content of blood is listed here. In men, it's 13 to 18 grams per deciliter. Women, 12 to 16 grams per deciliter. You don't need to memorize those numbers, but just so you have an idea about what kind of range of values we're thinking about. Okay. So we can get iron from our diet, but we'll also see that we are able to recycle some of it as we destroy and then make new red blood cells, which, as we talked about on Wednesday, we're kind of always doing. The other 2 things we're going to need from our diet are folic acid and vitamin B12. So these might be things you take supplements of. Folic acid, they specifically tell pregnant women to take supplements of. Both of these are going to be necessary for DNA replication, so making more DNA, which is a fundamental step of making new cells at all, any kind of new cells, right? Including red blood cells. So they're eventually going to lose their nuclei, lose their DNA, but to make new red blood cells, we are going to need to start out back at the top of this, right? We are going to start out by doing mitosis, making replicated cells up here at the top of the pathway that have all the usual cellular components before they lose them. So we need folic acid and B12 in order to do that process, in order to make any new cells at all. And then iron is specifically for the hemoglobin inside the red blood cell. Okay. When we are done with our red blood cells, remember that they have a relatively short lifespan. We're going to filter them out and we're going to destroy them. This is going to happen in the spleen. So the spleen is going to be filtering basically all your blood with the purpose of taking out old red blood cells. And it's then going to get help from the liver to break them down. So we're going to track through the waste components that this produces. We're also going to be taking a close look at what's happening with the iron which we're going to be partially recycling in order to keep making more hemoglobin. Okay. So basically, the spleen has macrophages in it, so that's a cell type. Macrophages filter blood by phagocytosis, right? So they're eating old, fragile red blood cells that aren't working so well anymore. Hemoglobin gets catabolized, so hemoglobin gets broken down, and we are going to rename, right, the molecule after we start to break down the hemoglobin. So the first thing we're going to do is we're going to remove the iron in the heme, and we're going to convert our hemoglobin to bilirubin, right? Bilirubin is going to be released into the bloodstream. It's going to travel to the liver, where it's going to get further metabolized. But then the products of breaking down the bilirubin are going to get secreted into bile. So bile comes out of the liver. We'll see also sometimes it's stored in the gallbladder. But eventually bile travels to your intestinal tract and gets excreted that way, basically in feces. The other place it can go is it can be released partially into the bloodstream, and it can be excreted in urine, so through the bilirubin catabolism. Fun fact, bilirubin has color. It's part of what turns feces brown. When you see it in the cadavers, it'll be green initially, but as it gets added to the other contents of the intestinal tract, that's what's creating that color. Okay. So we care about this largely because we care about recycling iron. We're recycling it so we can make new hemoglobin. Really care about hemoglobin because it's carrying all our oxygen for all our tissues. Okay, so a couple compounds, molecules that we want to know about as we track what's going on with our iron are transferrin and ferritin. So try really hard to associate these in your mind with iron. Right, so transferrin, ferritin. Okay, so these are going to be associated with iron. Transferrin is basically going to be carrying it around, and then ferritin is going to allow us to store it. And the thing that's helpful for remembering this has to do with iron. I think this FER is some Latin word that means iron. Sorry, my Latin's super rusty, but you'll know it's in both of these words. Okay, so our transferrin is going to transport the iron in the blood. So we're going to see iron being transported in two cases, basically. So this first one, from the GI tract to the bone marrow, that basically is referring to dietary iron, right? So if you eat something that contains iron, you want that iron to make hemoglobin for your red blood cells. So it has to get out of the GI tract, has to be carried through the blood to the bone marrow. So transferrin, in that case, would connect up, bond to this dietary iron, carry it to the bone marrow where we're making our new red blood cells. The other place we're going to see transferrin is going from the liver to the bone marrow. And this is because our stored recycled iron is going to be stored largely in the liver. So when we see transferrin going from the liver to the bone marrow, basically what's going on is it's picking up iron that we stored for later. So taking it from its storage site in the liver to the bone marrow where we are making our new red blood cells. That storage is happening by binding iron to ferritin. So in the diagram that we'll look at in a moment, you're going to see things labeled T iron and F iron. What that means is iron bound to transferrin versus iron bound to ferritin. That's what they're going to be talking about. So we'll see it stored bound to ferritin, mostly in the liver, though sometimes we'll see some of it in the spleen and some in the small intestine. Okay. So we're going to walk through this big picture. This is showing us basically the whole life cycle of our erythrocytes. So the whole life cycle of a red blood cell. We're going to break it down. because as usual, it's a pretty busy diagram, but this is where we're going. So we can see we have our bone here. We got a femur. We can see we got some bone marrow. We see blood cells coming out of it, right? Those red blood cells. We see them getting sucked into the spleen. And then we know some components are going to go to the liver. We know the GI tract is involved because that's also part of where we're getting iron, right? Our dietary iron. So that's on here. And we can see the kidneys because we remember that the kidneys are sending that signal, that erythropoietin signal, in order to tell us to make more red blood cells. So that's just tracing through the big organs and bones that you can see in this process, and we'll talk through the molecules next. Okay. So here we have our femur, just an example bone. Femur has bone marrow. This is the site of production of our erythrocytes, production of our red blood cells, right? So we can refer to that as erythropoiesis when we're making red blood cells because it makes erythrocytes. Remember that the signal for this is coming from the kidney. We'll see it at the end of the picture, but that erythropoietin is telling us to make red blood cells. Okay, so our red blood cell circulates around. So it lives for a while, right? It doesn't directly get murdered by the spleen, but eventually, when this red blood cell becomes old and fragile, it's going to get pulled into the spleen, and that erythrocyte is going to get broken down. So the first step that the spleen does is it breaks it into the heme group. So remember, that's the part where we have the iron, and the globin group, which is just some of the rest of that polypeptide chain, right? And then those two things get further broken down. So the heme group specifically, which is what we really care about for the purposes of this class, right? So focus on this left-hand side of this equation. The heme group gets broken down into iron, super important. We're going to need to recycle that, and bilirubin, which is going to be a waste product that is going to eventually get excreted, right? Okay, so the heme group, we're pulling out the iron, we're breaking it away from this other piece, the bilirubin. So that iron is then going to enter the bloodstream, bound to transferrin. So that's what we see here, this T iron. So this transferrin is kind of like a car, right? It's transferring the iron, okay, T iron. The bilirubin is also going into the bloodstream, but just kind of on its own. Amino acids, after we break down the globin, go along with it. That part's less important. So focus on what's going on with the iron. Okay, so heme, broken down iron into bilirubin. Iron gets bound onto transferrin. Next, we have the liver. Okay, so the bilirubin is going into the liver and it's being converted into bile, which can go into the bloodstream, also can go into the gastrointestinal tract. We'll also see some of that T iron. The air doesn't kind of go all the way there, but they're all kind of going together. Some of that iron is going to get to the liver to be bound as F iron. So that's the iron bound to ferritin. So that's like our storage of our recycled iron. Okay. What this is also showing now is it's also on this diagram, we have like the production portion of the erythrocyte life cycle. So that's why we see the T iron coming out of the F iron here. This is telling us that we will also start to release iron from the bound form of the iron, the ferritin iron in the liver, when we start to make the erythrocytes. So the reason for some of this messiness is because we're showing both at the same time happening. Okay. So here we see bile, some of it going into the bloodstream, lots of it going into the gastrointestinal tract is part of why we see that change in color in the feces, right? As the bile goes into the small intestine, large intestine. We'll talk about the intestines later. But the other thing that's happening with our gastrointestinal tract is we see the dietary iron coming in. So we have iron coming in from ingestion, right? and the cardiovascular system, right? The small intestine, right? It has blood vessels, it has capillaries right along it, just like everywhere else in our body. So that iron that we're eating as it goes through gastrointestinal system makes its makes its way into the bloodstream, again bound to transferrin. So we got that T iron. Okay, so we have T iron circulating in our blood at this point. Some of it is from breaking down the old blood cell, right? Some of it is iron we may have released from its storage site in the liver where it was bound to ferritin. Some of it is dietary iron. So we got this T iron circulating around, which brings us to the kidneys. Okay. So one of the things that's happening in the kidneys is the bile that we put into the blood. So the part that didn't go into the GI tract, it's going to go through the kidneys and get excreted in urine. So part of that is leading to the yellow color, right? The other thing that the kidneys are doing, more important, I would say, as related to the life cycle of the red blood cell, is they're giving off the erythropoietin, telling us to do erythropoiesis, which is the creation of new red blood cells, the erythrocytes. So that is really the whole life cycle here drawn on. Okay, questions about this before we talk about a few last topics? Okay, cool. Okay, so one of the interesting clinical connections to our understanding of how these erythrocytes get produced is anemia. So anemia is basically telling us that we have not enough hemoglobin, right? Which is often meaning not enough red blood cells. So we have a number of different types of anemia coming from different parts of this production process that get broken. So dietary anemia, the one that's caused by what you actually choose to eat, is going to be caused by iron deficiencies, right? If you're not eating enough iron, or deficiencies in vitamin B12. Now, vitamin B12, when you have this specific type of dietary anemia, is called pernicious anemia. It just has its own special name, but it's a form of dietary anemia. Hemolytic anemia can be caused, or has two types, basically, malaria or sickle cell anemia. So this is going to cause poor oxygenation because of actual damage to the red blood cells. We can also have aplastic anemia caused by bone marrow defects, renal anemia caused by kidney disease, hemorrhagic anemia caused by bleeding. So, but all of these are fundamentally going to have the same effect. We're going to not have enough hemoglobin, not have enough red blood cells circulating around in our bodies in order to effectively oxygenate our tissues. So that's why we care about anemia. Because of the, yeah. Okay. Let's see if I have this as a poll question. The development. Okay. So just a quick reminder. Okay. So we're going to close this poll. Okay. So most people got that it was erythropoietin. Erythropoietin is made by your kidneys. So it's a type of cytokine called a hematopoietic growth factor, and specifically, it's the one that tells us to make red blood cells. So there are multiple. They control overall all kinds of blood cell production, but this is the specific one that creates the red blood cells. Okay. So here we got a question about our life cycle. Looking great. I'm going to end the polling. Yeah, they're born in the bone marrow, right? Produced in the bone marrow. That's where we start to make them. And then they die when they get filtered out by the spleen, broken up into their heme, their globin parts. And then the heme gets broken into the iron and the bilirubin. Okay. So quick, I guess, announcement this is, we are actually going to skip this section on leukocytes, at least for now, for two reasons, for time reasons, and also because we already covered this in Phys Lab in ANP1. So this is kind of a repeat of what we did at that point. If you guys really desperately want to go over this stuff, let me know and we can go over it on our review day. But for now, we're just going to cut this, which I think people can appreciate a couple fewer topics on the exam. Okay. So I'm going to click through this section on our different types of white blood cells and when they become important, if they become important in any of our future units, we'll do a quick review of them. Okay. So this is so we can have time to talk about platelets and hemostasis. Note that this is not homeostasis, slightly different word here. Can be kind of tricky because they made it look almost the same. So hemostasis, which means maintaining the blood, right? Rather than maintaining just any sort of regulated variable. Okay. So hemostasis is really important because you want to not bleed out, basically. So that's the whole point of the process that we're going to talk about. It's going to start with platelets. So we're going to see that in order to maintain your blood and not bleed out after an injury, first what you're going to have happen is that your blood vessels are basically going to constrict, reducing blood flow to the area. You're going to use platelets to form a platelet plug, which is like kind of a very temporary kind of Band-Aid. And then we're going to form a blood clot at that location, kind of supplanting the platelet plug. And we're going to talk through all of that. Okay, so platelets are the beginning of this process, basically. Okay. Right before we have the platelets get involved, the immediate reaction to an injury is going to be something called a vascular spasm. So vascular, just referring to our blood vessels, spasm, literally like. So when a blood vessel gets damaged, it's going to immediately constrict. Remember that when something constricts, right, it decreases the radius, increasing resistance, meaning the flow to that blood vessel is going to decrease. So that's the point of the vascular spasm. It is an intrinsic vascular response, so it is just something your blood vessels do on their own, but sympathetic innervation also does come into play. Remember, our blood vessels have all of those sympathetic nerves going to them, right, that can release norepinephrine. When norepinephrine releases, it binds to alpha receptors. In the smooth muscle, they constrict, causing vasoconstriction. So that whole process would be occurring here as well. Okay. The other thing that's happening at this very initial point is that the endothelial layer is going to start to become sticky. So I'm going to pull up a picture real quick to kind of give us an overview, like visually, of what we're talking about before we go through the words on your slides, because I think it's kind of easier to see when you can actually see it. Okay, so what we're talking about when we're talking about the endothelial layer, right? So we have our like epithelial cells, basically those endothelial cells that are lining our blood vessels, right? They're everywhere, right? We talked about in capillaries, how we have continuous capillaries, we have fenestrated capillaries, but we always have this layer of cells, specialized epithelial cells called endothelium. When you have an injury, right? Right, so say someone stabbed you, right? So we had an injury coming here. This is exposing tissues below that endothelial layer to the blood in your bloodstream, right? So underneath that endothelial layer, right, like kind of out towards the tissue, we had like collagen fibers and stuff. It's been a while since we talked about extracellular matrix, right? We talked about that in Phys 1 lab as well, right? But we got some stuff under that cell layer. And when it's exposed, it's going to cause some reactions in the blood, basically. So that's what we're talking about. So when we expose these collagen fibers here that are below our endothelial cells, we're going to see that they get sticky, cause platelets to stick to them. Then the platelets themselves get sticky, causing more and more platelets to stick in that spot. So this is the formation of that platelet plug, kind of like a bird's eye view. We're trying to create this lump of platelets at the spot where we exposed some collagen fibers. And we'll look at the right side of that drawing when it comes back. It actually is in your slides in a minute. Okay. So we're forming the platelet plug. We can also refer to platelets as thrombocytes. So platelets are basically cell fragments, little pieces of cells. They're in that big chart of all the blood cells that we looked at a while back, and we looked at on Wednesday when we were looking at how the red blood cells differentiate. But basically, platelets are just colorless, tiny cell fragments, no nucleus. They do have some organelles and some granules. You have somewhere between 100,000 and 500,000 in your blood. They're important for blood clotting because they're this first step, right? Depending on whether we're counting from vascular spasm or whether we're counting from our first chemical reactions. But they're one of the initial steps of forming a blood clot. Okay. So the platelets or thrombocytes are starting to stick together around the site of the vessel damage. So this decreases blood loss, right? Because you basically have some platelets covering up the hole. So we decreased blood flow to the area with our vascular spasm. Now our platelets are actually trying to cover over the actual opening on the blood vessel. Okay. So this is part of our initial response. A platelet plug is not very strong though. So in order to really heal, in order to really effectively close this vessel damage off, we're going to need to form a blood clot, which will eventually take the place of the platelet. But we have to form the platelet plug first. Okay, so if we want to see this as a flow chart, we can look at it like this. So we have our blood vessel damage exposing the subendothelium. Okay, so sub, below, endothelium was that layer of cells that line all of our blood vessels. So this is referring to that layer with the collagen fibers that's basically on the base of that layer of cells. Okay, so that's what we have happening first. Then we start to have the stuff where the platelets start to stick together. What is actually happening at the very beginning that wasn't written in on that image is we have something called von Willebrand factor. So von Willebrand factor is this molecule that directly binds to the collagen fibers. And the von Willebrand factor is kind of sticky, right? So when we said the subendothelial layer gets sticky, when we said that collagen fiber gets sticky, part of what we were talking about really was this von Willebrand factor reacting with the collagen fibers. Okay. Platelets bind onto the von Willebrand factor. We then have more platelets stick to each other. We have the platelets stuck to the von Willebrand factor and therefore stuck to the subendothelial layer, stuck to this collagen fibers. The platelets are also going to secrete some different chemicals that are going to basically cause positive feedback, so causing more and more platelets to stick together. So we will now take a look at that. Not that drawing again yet. We're gonna talk about aggregation. So just note here, difference between adhesion and aggregation. So adhesion here is talking about like that initial sticking of the platelets to the site of injury as they bind to this von Willebrand factor. Aggregation is talking about the platelets sticking to each other, forming a larger and larger plug. Right. So the aggregated platelets, so as they're this bigger plug, they're releasing some more chemicals. So that's what we're referring to with these secretory products here. So they release something called ADP. ADP increases the stickiness of the platelets that are there, sticking more and more platelets to it. Serotonin and epinephrine, which we've seen before, kind of cool that they come back here in our clot formation, right? Serotonin and epinephrine are going to cause more vasoconstriction, so kind of building on that reaction from the vascular spasm. And then we're going to have other chemicals that are going to facilitate blood coagulation, aka our formation of the clot, which we're talking about next. They also produce this chemical called thromboxane A2, which, like ADP, is kind of doing positive feedback. So it's doing a bunch of these things as well. It's also increasing the stickiness. It's also helping with the vasoconstriction and so on. Now, the thing we want to be careful about as we're forming a platelet plug, right? We want to form our platelet plug and then our blood clot just at the site of injury, right? We don't usually think of blood clots as a good thing. And the reason for that, right, is that they cause a lot of problems when they form where they shouldn't, right? If you have blood clots going just through your arteries, getting to your heart, that's a problem, right? If they get into your brain, right, they're going to cause a stroke. We don't want clots just forming everywhere. We want to really, really carefully regulate them. So the platelet plug is going to be kind of hemmed in by some molecules that are created by healthy endothelial cells. So the cells that didn't get damaged, basically. So the cells that didn't get damaged are producing two things. They're producing prostacyclin and they're producing nitric oxide. So these two things are basically stopping the platelet plug from just kind of getting sticky in one spot and then aggregating more and more platelets all along the blood vessel, right? They're only able to stick at that spot because everywhere else we're releasing these other two chemicals saying, don't stick to me. So coming back to this picture, so we see on the left-hand side, we can see our adhesion and aggregation, right? So we exposed our collagen fibers in the subendothelial layer. I'll pull up my pen while I'm talking, right? Just directing our attention. So we exposed collagen fibers in the subendothelial layer here. Our platelets adhered to them. So they initially stuck to the collagen because of von Willebrand factor. Von Willebrand factor, which is not written in on this particular diagram, but that was the initial chemical that basically made this collagen sticky. Okay. Then the platelets aggregate, right? So they get stickier and they stick to each other, right? And that is largely because of ADP. So one of the things they're producing is ADP. It makes them stickier and it makes more platelets stick to them. The other thing they're producing here is our thromboxane A2, right? TXA2, which is also fundamentally making them stickier. What it actually does is kind of make us make more ADP, but that part's not important. We can just kind of lump these two things together for our purposes. So that's the aggregation part of our platelets, right? So we're forming our platelet plugs. So far, so good. We'd also be giving off serotonin and epinephrine, causing more constriction of the blood vessel. As we move over here to the right, we could see what will be happening in this neighboring cell, basically. So think of the right side as like zoomed in on here, and then on the cells to like this side. So in order to not have this platelet plug spread out like that way and that way, these neighboring cells are going to be giving off our two chemicals that prevent platelet aggregation. So prostacyclin and nitric oxide. So as the platelets maybe try to stick over here, right? They're going to try it. Can't do it because of these two chemicals. So they are inhibiting platelet formation over here. Okay, which brings us to clotting and the formation of the blood clot. So clotting can also be referred to as coagulation. And we'll see that we have a bunch of coagulation factors. We're going to have a lot of molecules associated with this. But fundamentally, what we're trying to do here is we're trying to convert our blood into a solid gel, which we call a clot or a thrombus. Okay. It occurs after we form the platelet plug, and it's going to be stronger, basically. It's going to work better to block off that site of injury while the cells heal. And this is our dominant hemostatic defense mechanism, meaning this is the most important part, basically. So we are about to see a bunch of scary diagrams. So before we do that, I'm going to orient you to the very basic, most important part of what we're about to talk about, which is that what we have happening to form this clot is we're following this molecule fibrinogen, which is circulating around in our blood. We're going to convert it into something called fibrin. So this is like an activated form of fibrinogen. Right, so we're essentially activating fibrinogen, creating loose fibrin, and then we're going to further convert that fibrin into a stable mesh. So we have our loose fibrin, and then we have our fibrin mesh, which is stable. We're going to talk about a lot of factors that like come in here and convert fibrinogen to fibrin, right? So that activate it. We're going to be talking about other factors that come in and help us convert our loose fibrin to the fibrin mesh, which is why the diagrams start to get kind of overwhelming looking. But if you have to back up and go back to the basics, the really most important part is that we're trying to make fibrinogen into this stable mesh. So just these two steps here. So that fibrin clot is our fibrin mesh here. That's our blood clot. That's the whole point of everything we're about to talk about. Okay. So real quick, before we move into the clot formation. So how did we get to actually starting to form the clot? Great. Yep, I see people answering right. Right. Our platelets are adhering or sticking to that subendothelial collagen. They're then aggregating, so they're sticking to each other, forming the platelet plug. And then we're going to be forming this fibrin clot. Okay. So I'm going to highlight for us here what we just talked about. So part of this that should be familiar from the previous slide is down at the bottom, right? So we're talking about things that affect how fibrinogen gets turned into loose fibrin and then into a stabilized meshwork. So this was that most important part that I was telling you about in our previous slide. A little more orientation here just before we kind of dive in. So the way this diagram works is when it shows you something highlighted in like the lighter pink, it's telling you that this is something that was like circulating around in the blood, but was kind of inactive. So we have our fibrinogen, we got this prothrombin thing, we got inactive X, right? So that's one thing that's going on here. The other thing that's going on is that we see a lot of words, right? But we'll see that we have like our inactive form on the diagram, and then we have our activated form, right? So these are the same thing, but this is just telling us that we had it already there, and then we activated it with a specific mechanism. So up here we have our intrinsic and extrinsic pathways. We'll talk about them in a bit. Okay. So hopefully that can make this a little less overwhelming, because we don't really have as many words to remember as it looks like we do, because they've written out both the inactive and the active forms. Okay. So next piece. Okay, I'm going to talk through this here. So basically, we're interested in how fibrinogen turns into fibrin, and then how that becomes a stabilized meshwork. So we're going to be tracking first, okay, what lets us do this first step, right? What lets us turn fibrinogen into fibrin? And the answer to that is that this molecule thrombin lets us turn fibrinogen into fibrin, right? So thrombin is the molecule turning fibrinogen into fibrin. And thrombin itself is the activated form of something called prothrombin. So we see prothrombin there. When it's activated, creates thrombin, which then allows us to convert fibrinogen to fibrin. So when you're looking at any of these diagrams, I actually kind of recommend going from the bottom up, right? Focus on this most important part, and then kind of move backwards to see how these things affect it. Okay, so fibrinogen is turning to fibrin because of thrombin, which came from prothrombin, which came when we had this coagulation factor X. Okay. So this is like the flow chart of why we are able to convert fibrinogen to fibrin. The other thing that we can see here on this diagram are the things that are affecting, and I'll pick a new color. It's a good color, blue. Okay, so the other things are the things that are affecting the conversion of this loose fibrin that we start out with into our stable meshwork, right? So what affects this second step? We're going to see what affects the second step is something called factor 13. And very unfortunately, they numbered all these coagulation factors in the order they discovered them, not in any order that is actually present in their pathways. So try to think of these as just names as opposed to numbers. It's kind of helpful for that reason that they're in Roman numerals rather than, our numbers. Okay. So this factor 13 is helping us create this stabilized meshwork. Oh, and we see that thrombin had an effect on that as well, right? So there was an extra step in between, but basically thrombin activated this factor, which then stabilized the meshwork. Okay. So that's how we want to read this diagram, basically. Having walked through that, we can see that this factor X is really important. So this factor X, essentially, its role is to turn prothrombin into thrombin, which then works on both of these like parts of our really important pathway, right? Thrombin directly converts fibrinogen to fibrin and indirectly converts our loose meshwork into a stable meshwork, right? Indirectly because we have this extra step in the middle, this factor 13. So of our factors that we're going to have on here, the ones that are numbered with Roman numerals, if you are going to remember them, focus on remembering factor X, right? So factor 10, our most important of the factors. We're going to see more of them than just the ones on here. But factor X is really crucial to this process. Now it starts to look scarier because we zoomed out farther, but we can see that the whole bottom of this diagram is just the same as what we were looking at, right? This is like we copy pasted what we just saw on the previous slide here on the bottom, right? So here's the super important part we've been looking at. We're focusing on fibrinogen to fibrin to the stabilized meshwork. It's our very last line. And then we looked at this cascade from factor 10, factor X, right? So this inactive X converted to X, whatever, right? We have this active form. You can just kind of lump those together here. So factor X converted prothrombin to thrombin. Thrombin then directly allowed fibrinogen to become fibrin, and it indirectly allowed it to become a stabilized meshwork because it activated this factor 13. Okay, so this was the fundamental piece that we just went over. So what we're seeing now here that's new is that we have both intrinsic pathways and extrinsic pathways that lead us to the activation of factor X. Okay. So what we mean by this basically is that some of the ways that we activate this factor X are going to come from the blood vessel, right? So the damage to the blood vessel itself, that's going to be this intrinsic pathway at the top. Other factors are going to be because we actually basically, right, to damage a blood vessel, almost always the tissue surrounding that blood vessel is also going to be damaged. So we're going to have factors, coagulation factors, extrinsic to the blood vessel, so in the tissues, that also activate this factor X. So these factors in the extrinsic pathways and the intrinsic pathways, their numbers, please don't worry about memorizing those. I think that this is a complex enough process that I want you to focus on what's going down here in this blue region. So what I want you to know about the intrinsic and extrinsic pathways is basically that the extrinsic pathway is coming from damage to the tissues. So you could remember the phrase tissue factor, but don't worry about the numbers here. Okay. So if we step through the intrinsic pathway, what we can basically see is that the intrinsic pathway, it's intrinsic, so I'm saying it's coming from damage to the blood vessel directly itself. So it's coming from contact with collagen, basically. We have a cascade of coagulation factors. We're not worried about memorizing their names and numbers, but they exist. They eventually activate this factor X. We also have this extrinsic pathway. It involves tissue factors, which are produced by damaged tissues surrounding the blood vessel, also activates our factor X. So they come together, and then they do the things we talked about in the first image. They turn prothrombin into thrombin. Thrombin then gets to convert fibrinogen to our loose fibrin. And thrombin also activates this other clotting factor, so it indirectly converts fibrin into its stabilized meshwork, right? So there's this intermediate step, but essentially, if thrombin isn't there, this isn't happening either. Okay. All There are a couple more slides, but I know we're at time. So we'll cover the very end of this blood clot stuff on Monday before we do our review. There's not a lot more. We got to the big bulk of it today. So this is just some kind of clinical stuff really is what we got left here. All right, everybody have a great weekend. Yep, what's up?
How do you intend on doing review days? What's the way you do review days, I guess?
Oh, okay. Yeah, so for those of you who didn't have me last semester, if you have questions that stuff you want to go over immediately, great, come with those questions, we'll go over them. If you don't have direct questions, I like to come up with like a short worksheet with some sort of problems that allow you to think through a bunch of the topics that we've been going over. So they're not like multiple choice like they would be on the exam, but they can kind of jog your memory on what topics you don't understand and what you'd like to talk more about. So we kind of go back and forth between working on those worksheets kind of in groups, and pulling back, seeing what questions came up for everybody. Then we can talk through those topics in more detail and kind of back and forth like that. Does that make sense?
Awesome, thank you.
Yep.
So I just want to make sure I didn't miss anything. There's no quiz this week because we have that exam next week.
There is a quiz on this final set of materials so that you have practice.
That's due on Monday?
Yep, due on Monday.
Okay, perfect. Thank you so much.